Good evening. My name is Nanthi Bolan. It is my pleasure to welcome you all to this special lecture this evening. Now, this is the lecture today, and before we start the lecture, it is customary for the University of Western Australia to acknowledge that the university is situated on Noongar land, that the Noongar people remain the spiritual and cultural custodians of their land and continue to practice their values, their languages, their belief, and the knowledge. Now, on behalf of the University of Western Australia, I wish to warmly welcome you all to this lecture by Dr. Mark Farrell on the challenges of managing nitrogen and organic matter in dryland systems. Now, Rosanna has prepared these dot points, so I have to follow strictly the protocols. Now, Dr. Mark Farrell is a principal research scientist at CSIRO. There are some ex-CSIRO great scientists are here. And Mark leads the Biogeochemistry team. Mark holds a BSc Honours and PhD from the University of Wales, England, in the UK. And more importantly, he is an adjunct professor at University of Western Australia, School of Agriculture and Environment, where myself, Lynn, Louis belong, many others belong. And Dr. Farrell is a biogeochemist with expertise in interactions between organic carbon, organic matter, and nitrogen in soil, and the importance of these on ecosystem function and agricultural productivity. He uses radioactive stable isotopes to track the fluxes of these elements, carbon and nitrogen, through the soil, plant, and microbe interface. And he also uses spectral techniques, including NMR, MIR, to investigate the chemistry of these element pools in these ecosystems, including agricultural ecosystem. Dr. Mark's research spans from fundamental work to identify and isolate important functional processes through to broad applied science. He's working for CSIRO, so they have to do a lot of applied science, outreach activities, where outcomes are deliverable to partners or end users in the industry and the government. He has studied many different land users and ecosystems within Australia and internationally, and he seeks to find some commonalities and differences in these processes and function between these ecosystems. Why I am introducing? Personally, I have supervised a PhD student with Mark when I was at University of Australia. I must tell you that Mark is an excellent supervisor and great mentor to PhD student. That particular PhD student published more than 7 papers within the PhD, with Mark as the main author. So now I welcome Mark to give his presentation. Thank you.
Thanks very much, Nanthi. Thank you for the introduction. And most of all, thank you to you all for coming and listening to me. I hope I don't disappoint. I've been G'd up a little bit by at least 1 or 2 people on social media, so I shall try my best. So yes, challenges of managing nitrogen and soil organic matter in dryland systems. That's certainly a challenge. And first of all, because almost certainly I'm going to talk for longer than I really need to, I'm going to get the important things out of the way first. None of the work I'm going to present would exist without generous funding from several funders. I think that's all the relevant ones for the slides I have in here today. The usual suspects, various Australian federal government departments. We had some really great work done in cover crops, thanks to the National Land Care Programme, and perennial and very important sponsors, GRDC, who fund an awful lot of process-based research, as well as applied outcomes in the country. And then on the right-hand side, I'm very much standing on the shoulder of giants, at least one there, Jeff, top left, Jeff Baldock, who retired from CSIRO a couple of years ago, long-standing mentor of mine. Other colleagues at CSIRO, Lynn McDonald and Sanani Karunaratne, Uday Nidamolu, and then outside of CSIRO, Naomi Wells and James Hunt, both excellent emerging scientists. And just to listen to all of us as scientists, none of us would have any data without the Rose Gallery at the bottom, Athena, Tom, Janine, Steve, and Paul, who process the samples in the lab and actually generate the data in the first place. So, let's have a go at this. Soil organic matter. What is it? Well, tomorrow I have a workshop with students asking just that question. So I'm going to have a go, first of all. I may upset 1 or 2 people with the next couple of slides. Traditionally, we have seen organic matter as these things: humic acids, fulvic acids, and even humin, principally defined by their chemical recalcitrance in soils. I'm not even 40 yet. This is what I was taught at university too. So I'm just raising this as a point of how much things have changed in the last 15 or 20 years, because I think it is quite important to understand this. These fractions were basically got at by some very agricultural bucket chemistry. Dissolve in a strong alkaline, precipitate in a strong acid, do things gravimetrically. The world, since I did my PhD, has quite literally turned on its head in this space. And we now principally understand organic matter to be defined by the soil environment and principally biological reactions going on within it. And that organic matter is actually protected from decomposition rather than being recalcitrant. And that's also an important point to bear in mind. It is also presumed primarily to be microbially derived. Now, that goes back a long way. David Jenkinson's out of Rothamsted, it's his famous line from the 70s about everything going through the eye of the microbial biomass needle. But again, all these things point a little bit away from this principle of condensation and other abiotic reactions that got you this stable stuff. So that's the contentious bit out of the way. I do occasionally get some vigorous debate on that topic, and I think there is some. I should emphasise. I think there is room for both of these concepts here, but I would argue strongly that it is the new version on the right that is the principal driver, and that some of these perhaps solid-state reactions in the soil could be happening, but they happen at a very slow rate over a very long period of time, and by that matter alone, can't possibly be the dominant reason by which organic matter is stable. So that's what it is, or what I think it is at the moment, and that might change by the end of the presentation. I'm now going to go through how we actually go about looking at it. And this is the fractionation scheme that was pioneered by my colleagues at CSIRO, actually before Jeff, mostly it was John Skemstead who really was putting this together. And emphasizing the important bit here that soil carbon, we typically accept anything bigger than 2 millimeters as not even being soil, and that's contentious. Becoming more contentious in itself, very operational, definitely comes from soil texture classes rather than having any biological relevance to it, and there's plenty of good work being done in Western Australia in particular at the moment on gravel soils, demonstrating that gravel is actually quite important. But nonetheless, those are long-standing conventions, and these two fractions here, particulate and humus-like organic carbon, now the Americans call the humus-like organic carbon mineral-associated organic carbon, which I think is probably a more accurate term for it. We follow down this so far. There's a pretty straightforward continuum. Carbohydrates decrease, carbon to nutrient ratio decreases, and resistance to decomposition increases. Now the joker in the pack is resistant organic carbon or charcoal-like material, of which we have plenty in Australian soils. And that stuff lasts for millennia, which is why I bring it up separately. Yes, it's carbon. Yes, it counts from a carbon sequestration perspective. And it's going to stay there for a long time unless it blows away or erodes somewhere else. But it's not providing the same ecosystem functions that we tend to think of organic matter doing. It is behaving probably a little bit more like a clay particle. And these fractions look different when we look at them under an electron microscope. Particulate, like here, you can just about make out it still looks like bits of broken up plant material. Whereas the humus-like carbon is much more waxy. And that, amongst other things, has led us to believe that principally this stabilized humus-like carbon that's associated with clay materials is microbially derived. And using some NMR techniques to demonstrate just how different those three fractions are, you can see the HLC and the PLC are fairly complex, but they do differ quite substantially. And I'll come into that into a minute. The resistant-like carbon is very, very different. So those are the fractions, those are the tools that we use at CSIRO and many other people have used within the last decade or so since SCARP, a major project that was led at CSIRO with collaborators across the country, not least UWA. What does this actually tell us about carbon though? So we're trying to get back to what it's actually doing in the environment. Well, first of all, to get at those fractions, principally, as you may have spotted up there, it's a size fractionation thing, greater than or less than 50 microns. And that gets us to physical things that we can put through instruments and actually have a look at. And in the coarse fraction, the green here, you can see that there is a really wide difference in C/N ratio between the two. So it's very varied and generally higher. Whereas this fine material, which contains the humus-like carbon, is much more constrained. So there's something chemically different between the two, just on their C/N ratios alone. Bringing in some of the NMR data now, looking at the alkyl to alkyl ratio. So this is basically lipids to carbohydrates, long-standing convention that the greater this number, the more decomposed it's been. Again, the idea being that it's been decomposed by microbes and turned into lipids. Lipids themselves aren't particularly stable. They just stick to things, which makes them stable. And here is why it's important to consider the charcoal. There are other things that could be in the aromatic region of an NMR spectrum, but principally in soils it is charcoal. And there is no difference really between the two size fractions at that stage. And this is what causes us such a problem with what causes pretty much anybody who tries to do fractionation work a big issue, because it's small and it's coarse, it's light and it's heavy, the two ways we typically can go about fractionating things. But yet it's there, and it can be quite a large amount of it in your fraction. And if your fraction contains a lot of charcoal, it is not going to behave in the same way as you would expect from organic matter. And this is pretty systematic. So these are NMR spectra from, this is about 600 spectra in total, from SCARP. Coarse on the left, fine on the right, and then going down the depth profile. So this is, again, from the National Soil Carbon Programme that ran 10 plus years ago. And what's clear here is that there is a pretty systematic difference between that big peak on the left-hand side, so that's the alkyl or the carbohydrate peak, and the lipid alkyl peak on the right. Between the two, they're different, and with depth, that difference increases, particularly in the stabilized fraction. Again, really indicating that this is lipidy microby stuff. There are other techniques we can use to have a look at what this stuff is, though. This is some great work that's emerging from Naomi, one of my collaborators who I flagged earlier on. She's now at University of Lincoln in New Zealand. And she plays with stable isotopes. She's far better at stable isotopes than I am, and better at stable isotopes than most other people I know when it comes to natural abundance and interpretation. The principle here, I'm not going to go into too much detail, is ultimately stable isotopes tell us a little bit about what's going on in the soil. They don't tell us everything, they integrate lots of processes. But one thing to be fairly certain is that if there are no processes going on, or no processes that change things differently, then stable isotopes should be the same. And all those blobs should be pretty much on top of each other, and there shouldn't be differentiation between crop types, climate, or anything else. The very fact that there is on this figure demonstrates that fundamental things are happening in the soil donor profile to this material, and it is being changed by microbes, it's not being directly derived from them. And the last tool I'm going to talk about in trying to get our heads around what this stuff actually is are biomarkers, and in this case amino sugars. So these are a class of fairly conserved molecules. They're fairly preserved in soils relative to things like amino acids and phospholipid fatty acids that turn over quickly. So as we have different markers that are used for different groups that appear to be preserved from different groups, so bacteria or fungi, you'd hope that might be helpful, telling us whether or not it's bacterially or fungally derived, or by balance, whether or not it was plant-derived. But the trouble is those conversion factors, particularly around muramic acid conversion up to total bacterial biomass, are very woolly. And when you get back into the literature, they come from a startlingly small number of studies. I lied. There is one last thing, and I touched on it before, stoichiometry. The Redfield ratio, the idea that, well, in Redfield it was algae in marine systems, have a very conserved ratio of carbon to nutrients. Soils appear to follow this. This is data from a review that Clive Kirkby did, again, more than 10 years ago now. And some data, both from the literature, a wonderful study, Josiah Crane in 2015, showing that C/N ratio decreases with decomposition towards the fine fraction, so to delta 15N increases, and again, that little box and whisker plot that I demonstrated, showed earlier as well. So collectively, I can see how you can use all these bits of information, and that's convinced me fairly well that a lot of organic matter is dead microbe, but there could be a question. And there is a question. And this is why I said about turning the world on its head again, at least from an organic matter perspective. In the last 12 months, and indeed last couple of weeks, with this paper by Emily Waller out of the US, some really laser-like interesting discussions about whether or not these things are actually telling us what we think they are. Now, I've been chatting with Jessica Inokovic, who was actually a postdoc in our group until a few years ago. She's now associate professor at University of New Hampshire. She said that the main goal of that paper was actually to provide the answer, and they very quickly worked out they couldn't provide the answer. So all they've ended up doing is being in a really unfortunate position of being the negative Nancy, of saying, well, none of it really works that well, but we don't know what we can do about that fight. I think ultimately, all these tools can be ambiguous on their own. I think they're fairly ambiguous together. But if we can take them together, and all of them point to the same sort of direction, then perhaps we're actually getting somewhere with understanding what drives this. And this figure, which is, there are some excellent figures in that paper. This is one of the best, basically showing that most of these things that we would say come from microbes on the blue side, you can also find them on the red side direct from plants. And I will come back to the implications of that a little later. And just to highlight the opportunities that we have for trying to unlock that in this country, right now I'm leading the follow-up to the National Soil Carbon Programme, something called SOCM, Organic Carbon Monitoring. We're really imaginative with their names. This is only revisiting 300 sites, not the 4,000 plus that you can see there that Scott did originally, with a large number of collaborators. And principally, we're doing the same thing as Scott. We're looking at stocks, fractions, but we're also getting some other ancillary chemistry data to actually have a bit more understanding about what's going on and why. We're taking in some pretty detailed look at land use data. And because of that, and we're going back to sites that we sampled 10 to 12 years ago, we can obviously look at changes over time. And one of the opportunities we have by having this really valuable set of samples coming in is to be able to apply some of those tools I've just been talking about in concert rather than on their own, to really try and understand whether or not these fractions are properly telling us what we think they are. So I said I was going to talk about that in the context of Dryland systems. So this is a bit of a Jekyll and Hyde talk. We've had some really high-level chemistry stuff, and now we're going to go more to the applied side. So Dryland systems, I think we know mostly what they are. Principally, it's the fact that driven by rainfall, and in Australia, there isn't very much of it. Management, therefore, primarily is around conserving moisture so that there is enough available for crops within the growing season. They're not just an Australian phenomenon, obviously. To use my home state of South Australia and Goyder's famous line, this is work that UWA did a few years ago now, looking at the implications of different CSIRO climate projections on what would happen to Goyder's line, which, for those who aren't aware of what it is, it's basically this climatic boundary surveyed well over 100 years ago that suggested the limit of profitable cropping within South Australia. You can drive past the marker that proclaims where it is and carry on driving through cropping for some time at this stage, which is interesting. But nonetheless, and possibly one of the first bits of research that I read that actually demonstrated climate change is happening and is having an effect is this. And we know from modelling another work, this is turning it on its head, we know from modelling another work what the advances in agronomy and breeding should have got us in grain yields in this country. And the fact that they haven't moved very much can really only be pinned on the fact that it is becoming harder and harder and harder to grow them in that climate. So, what do we do about that? Well, this is ultimately best practice agronomy in Australian systems. I'm going to be slightly mischievous here and throw up a couple of slides I did for a GRDC talk a few months ago. Also, intriguingly, the main core tenets of regenerative agriculture. And I would argue quite strongly that a lot of best practice Australian farming really isn't that removed from the main principles of regenerative agriculture. And one of the things to bear in mind as growers in this country is an actual fact. You're well ahead of the curve. There are definitely improvements that can be made, undoubtedly, but the main tenets there and the main bonuses that are being spruiked in other parts of the world are coming from those countries adopting practices that we have followed in this country for probably 3 decades now. So what do these get us when we're talking about carbon? Well, minimal disturbance, bearing in mind the emphasis I put on the fact that organic matter is stabilised and not stable, minimising disturbance is actually pretty important. Maintaining cover, obviously it protects the soil from erosion, and it also acts to maintain moisture and carbon. Diverse rotations, again, good agronomic reasons for this, reducing disease pressure and spreading risk cross seasons. If your entire farm is planted to one crop and it's not a great season for that crop, you've got a problem. If you spread your bets, you have more options. Including livestock, and that's something I will come back to in a little bit as well. Definitely pastures input more carbon into the soil, that's undeniable. Pasture legumes are probably better at fixing N from purposes of a fallen crop, getting it as well, because of course a grain legume, you're harvesting the grain and taking away quite a lot of nitrogen in the process. And minimising chemicals. Well, that's the complex one. It's particularly complex when you come to fertiliser, bearing in mind that we're trying to grow as much carbon, as much crop as we can to fix carbon. Winding back the fertiliser isn't necessarily the answer. Another question that often comes up is, can we bring it in from external sources, of which there are plenty? Composts, manures, biosolids, biochars. Obligatory photo of a cow. When it comes to thinking about carbon sequestration, you need to bear in mind the concept of additionality. This carbon already exists. It's not freshly fixed carbon, so it cannot be counted. Any carbon, any extra carbon that the improvement in fertility of the soil, these things add, can be, but not the carbon itself. So if you add 1 ton of biochar carbon to the soil, that can't be counted as a carbon credit for you. The person who produced the biochar gets that carbon credit. But the real problem here is one of scale. And I have a postdoc who's working on biosolids at the moment, and actually did some sums at the back of a meeting he was hosting the other day. In 2021, apparently there were 349,000 tonnes of dewatered biosolids produced in the country, which sounds like quite a lot. But in 2021, according to the ABS, 32 million hectares of Australian land was under cropping, which means if you want to do it with biosolids alone, you'll be applying 11 kilos per hectare per year. So it can be important locally, but applying organic amendments is not a solution to the problem at the national scale. There just aren't enough of them, and that's before you get into issues of transport. So what can we do about it? Well, you're going to have to grow the carbon. And this is where there has been a little bit of debate recently in literature that I'll confess to taking part in. But nonetheless, the main tenet of this message is fine. Look, clearly, if the only source of carbon is coming from the plant, the amount it photosynthesizes and fixes is your upper barrier before you think about any of the loss pathways or anything else. The key thing to understand here, in moderating that message slightly, is that NPP now isn't necessarily what MPP might be in the future, and that can go both up and down depending on management versus climate and other stresses. So the strategies to increase fixed carbon in Australian cropping systems, I think, come down to 3 things. You can change the system, and by that I mean grow different things entirely. You can add to the system, try and grow some more things without displacing what you already grow. Or you can improve the system and, quite literally, grow what you grow better. So breaking down the different things, these are major system changes and are probably fairly unfeasible in a lot of cases, but nonetheless I think it's important to go through them. Agroforestry is one that gets talked about a lot, clearly with the industrialisation of a lot of Australian farming systems, getting large headers and seeders through a paddock with a load of trees. Now, how deep the rows just is a non-starter, and I accept that. But nonetheless, this is neat work out of France, showing that not only do you get an increase in the carbon from the tree itself, but also over time, a lot of those soil stocks have increased in the right direction too. So whether or not it's practical is a separate question, but at least in some places, it would have that effect. Then, what I think is more practical in our case is rather than trying to have crops and trees in exactly the same part of land, focus on sustainable intensification for the areas that are productive, and look at perhaps turning over some really less productive areas that you always struggle to get a crop over to environmental plantings. So that's the growing the different things. Growing more things, cover crops, and I've touched on this a couple of times, and I will give some more data on mine in a minute. There is very strong overseas evidence that cover crops, rather than having a fallow period, can improve soil health, various measures thereof, and particularly also soil carbon. That is not in doubt, and there are a number of studies like the one I have up here, covering quite large climate gradients in the Northern Hemisphere. There's another good one out of Europe that goes from southern Spain up to Scandinavia, all showing more or less the same thing. So the longer you have a plant growing there, the better. But, of course, this is Australia, where it doesn't rain very much. And the longer you have a plant growing there, the more it's transpiring, the more it is potentially using water. But nonetheless, with the idea of not displacing a cash crop, if you are in a wetter area that does receive sporadic rainfall in summer, it's not beyond possibilities. And this is the cover crops project that I was part of in South Australia. We had sites right the way across the southern region. Range of rainfall, annual rainfall from just over 270 at Waikerie, the tiny little blob, all the way down to nearly a metre more down in Tasmania. I don't have time to go into all the details of the study here, so I'm going to throw up a very rough summary slide. Basically, green is good, red is bad. All the sites are the individual rows. They're in order of rainfall gradient from driest to wettest. and then various factors that we measured across the top as the columns. So if you look at the far column on the right-hand side, which is the sum across all of there, it basically breaks it up into three different things. You've got the drier areas where cover cropping really did send most things backwards, and you've got to bear in mind that if you grow a cover crop that doesn't do very well and uses all your water, your cash crop isn't going to do very well and won't have much water either, and you actually end up with a net negative even for the soil itself because you didn't have a vigorous plant growing there at all if your cash crop fails as well. In the middle, it gets a little fuzzier, and I think that's really a more opportunistic opportunity rather than every year. But down at the bottom end, down at the wetter areas, and there are wetter areas in Western Australia too, there are fairly consistent positive effects from having had cover crops in. And I should emphasise, this project was probably one of the worst to try and get conclusive data from. It was a land care project. We had 20 sites on working farm paddocks. These were not on at experimental stations. This was done with with farmer scale seeders and headers. Some of the data that's missing, I'm missing some yield data from one site because the yield monitor wasn't turned on when the header went over it, for instance. This is about as operational as you can get for research. And we still found some fairly clear messages at the end. And one site I really want to show you is the site on Kangaroo Island. This is actually the result of only 1 year's worth of summer cover cropping, because it would have had 2, but there was a rather large fire on Kangaroo Island, unfortunately. And no cover crops aren't fire resistant. The green line, so the red here is the control with no cover crop. The green is a multi-species mixture of cover crops, and the blue, so the bar and the line on the chart on the right-hand side, single species. That's crop yield, and we had about a tonne hectare increase in oat yield after having had a cover crop in. So not a penalty to it, an increase in it. And really interestingly, given the fact this really is the effect of just 1 year's worth of change, a significant increase in carbon at depth in cover crops. So this is not inconsistent with a lot of the studies out of America in wetter areas there. It can work in wetter Australian systems. It's not a complete non-starter, but neither is it for everywhere. But I think that is, if we can do that in 1 year, that startled me. And trust me, I've checked this data a lot because I wasn't prepared to bet too much on it until I was comfortable that it is indeed real. So that's growing more. Now we get to the better crop question. And first of all, not quite crops. One thing to bear in mind is how much Australian agriculture has changed in the last 40 years or so, particularly moving away from having livestock in the system. So this is a study from Lindsay Bell and Andrew Moore from a good few years ago at CSIRO now, sort of showing those long-term trends in key states and also Australia overall. So on the left-hand side, proportion of farm area cropped, and on the right-hand side is dead sheep equivalents per farm area. And typically, cropping is going up and livestock are going down. So there's definitely, and one of the things I should add there is that a lot of the talk about organic matter levels dropping in Australian soils over time, a lot of it's talked about since clearing. I don't think it is since clearing. I think an awful lot of it has come from a move from pasture-dominated systems to cropping-dominated systems. And pastures are often forgotten as actually pumping a heck of a lot of carbon below the surface. So that's not even quite a crop. I'm going to post the extreme example now because everyone loves a picture of perennial wheat roots. Clearly, perennial wheat has got many problems, and I'm not an expert in this space, but there has to be somewhere some intermediate between the tiny little crops on the left, which are annual wheats, and perennial wheat. Surely there must be some medium that can be got at through crop breeding in there that gets you a resistant, high-yielding annual wheat that also puts more carbon below the surface than the paltry amount that our current elite lines achieve. And that, of course, might get you somewhere without ever having to change your system much at all other than sowing wheat, just a different variety. And it is being looked at, probably not as much as I would like. And no, I'm a soil scientist, not a plant breeder. That's definitely out of my field of influence. But whilst we're on it, better growth. Key one being overcoming your soil constraints. This is work from a colleague, Lynn McDonald, another GRDC funded project, looking at overcoming constraints to cropping in sandy soils in South Australia primarily. And at that Butte site, ripping got a yield increase of 1 tonne per hectare per year for 4 years, which is pretty impressive. But there are plenty of other sites up there where it did diddly squat. So you need to understand your soil constraint and address the right one. Don't just use a practice because it worked on that paddock. Then, better agronomy, basically. Better disease and pest management, improved varieties. If you can have a healthy crop growing vigorously and well, it will pump more carbon into the soil than one that's struggling with disease but you didn't want to apply fungicide. Now the biggest constraint of them all is nitrogen. This is a study from Rob Norton for GRDC back in 2016. 125 farms studied in southern Australia. Pretty much every single one of them was taking more nitrogen out at grain than was being applied by fertilizer. You cannot possibly build organic matter if you're taking nitrogen out of the organic matter to supply a crop. And yeah, that's basically the biggest driver of failure to meet water limited yield potential. So the question then comes, can we add more nitrogen? And I'll be up front, the slide was written before the current price of nitrogen. Nonetheless, it's still worth looking at. So there are risks. There are environmental concerns, and I'll touch on them briefly. Impact on the bottom line, however, isn't quite as clear-cut, or at least it wasn't until fertilizer ended up north of $1,000. This is data from James Hunt from a 5-year experiment. So that's your average partial end balance over 5 years on the x-axis, and your average gross margin on the y-axis. The R, by the way, is regional district practice, and then nil fertilizer right at the bottom. And this is James's nitrogen bank. I'm not going to go into too many details, but basically it's working on the idea of actually supplying enough nitrogen for the system rather than just focusing on the crop you might grow in that year. It's about providing enough nitrogen for the potential crop that could grow and recognizing that you may well catch a lot of that N that may not have been used in the following cycle. And basically ended up in a situation where the most economic on a 5-year basis is actually applying substantially more fertilizer than that regional practice, which is generally dialed back very conservatively to be on the cusp of maximal profit within a given year in a limited rainfall system, rather. And where this comes to ultimately is that agronomy is pretty much telling us that, yes, you can lose money if you apply too much fertilizer in a given year, but you forego far more profit by missing yield in the good years because there wasn't enough nitrogen in the first place. So, we managed to do all that, managed to do a nitrogen properly, do good agronomy, grow the right things. What happens? Well, Lynn and I came up with a fairly rough relationship between things that we can measure, a change in yield, and implying a change in carbon inputs. The greyed area is a review that John Sadaman did 12 years ago now, looking at measured changes in carbon as a result of improved farming practice, which is very vague terminology for Australian systems, but I suspect what he means is adoption of no-till. And you can see it's not really changed the dial much. Lynn's study from the subsoil constraints project I mentioned, that 1 tonne per hectare yield increase might be getting you an increase in upwards of 700 kilos of carbon per hectare per year increase inputs. And that's accounting for some decomposition too. We go to the nitrogen study now, and this is James's. So his best nitrogen rate versus nil, and then versus regional practice. Those lines are more or less of the same order as either. So applying more appropriate nitrogen rather than district practice, and improved practice. That's the same order of magnitude as Don Sandeman's numbers. Versus nil, bearing in mind that there are some advocates for really winding back fertilizer rather than applying it. Huge difference. The interesting thing is where the cover crops come in. The data from that Kangaroo Island site gets you all the way up there. when you count both the extra carbon input from the increased yield in cash crop itself, plus the amount of carbon from the cover crop. But back to the nitrogen story, it's actually fairly well established. If you want to build carbon, you need nitrogen. People who are advocating to cut back on nitrogen, and the idea that they're going to build a more resilient system, I think really do have the wrong side of the story. And if I can give one message tonight, that's it. It's not completely benign though, we know that. Nitrogen losses, of which there are many. Nitrous oxide is probably the more frightening of them from a climate perspective, although economic losses, even if you lose most of it, is fairly benign all the way through to dinitrogen. That's a pretty big loss, hit on your pocket at the moment. So we want to try and understand these losses and mitigate them as much as possible. Which brings us the challenge of ensuring adequate nitrogen supply. So roughly 1 tonne of wheat equates to an export of about 20 kilos of N. So 2 tonne crop, 40 kilos. But there are going to be losses. Losses you can do nothing about. There would be losses if you grew no crop there and fertilised. Nothing either. Organic matter still turns over and mineralises. Obviously a lot of the things that we do to it influence that, but nonetheless, it is mineralised. We're pushing for really high yields. Applying high rates of urea gets increasingly risky. And if you look at some of the high-yielding crop sites over in the east, you're looking at pulling off targeting 12, 14 tonnes of wheat per hectare. That's an awful lot of N to apply as urea in one season and expect most of it to stay there. So we need to look to incorporate more legumes and really look at fertilising the system over multi-years rather than the crop that you're managing in a single season. And we do need to better understand N losses, which is why probably some of the best news I've had this year was that the GRDC have invested 11.9 million in a new project that Louise is leading the UWA component of, I'm leading CSIRO's component of, and there's a good mixture of talent, organisations, universities across the country, really trying to finally get our heads around improving our knowledge of what's causing losses and understanding them better, testing this in the field and actually really getting our models up to scratch so we can predict them with a lot more accuracy, a lot more reliably, which can in turn help inform future management. So can it be done? Can we build organic matter in Australia's dryland systems? Well, there are some who'd have you believe you can't do it anywhere. I argued about that too, because we've known that organic matter mineralises for a very long time, and that was a very curious paper. Not least because David Paulson, one of the authors of it, was at Rothamsted, where the experiment to end all experiments continues to be done. So yeah, you improve, you add manure in this case. This is from Broadbolt, one of the classic experiments there. You eventually get to a point where you can add no more, but most of our farms, most of our soils in Australia are not there. If you stop doing it, yeah, you'll lose it again. This isn't a surprise. I focused a lot on the input side. You do have to understand that there are a lot of outputs. 2, the little rough and ready calculator that I showed does incorporate them. We're probably less pessimistic than the 10% argument of that Paulson paper, but nonetheless, we do consider them. And there's this big hairy beast called the Quarter per Mille initiative, or the 4 per 1,000, which is commonly misconstrued as thinking that we can increase our carbon by 0.4% per year. That's not what it's saying at all. It's 0.4% of what you already have. So in the context of a soil of 1.5% carbon, it's proposing you should be able to build it to 1.506 within 1 year, 1.512 in 2 years. Clearly, as you get further along, there's a compounding problem there, but ultimately, at least in principle, as a rough and ready idea, this was the aspirational target that was reached that is plausible, and a lot of us think it's plausible to some extent. That 1.5% example there actually really only needs you to be increasing carbon to meet the goal of 0.4% per year. You only need, in a soil with 1.5% carbon in the top 10 centimetres, you'd only need to increase your carbon inputs by an extra 80 kilos per hectare per year. So even if you assume that 90% of your biomass inputs get lost in the first year through mineralisation, as some of the more pessimistic views are, that's still only an increase of 2 tonnes per hectare per year biomass, which again sounds a lot till you look at what that cover crops example did. So in some areas it can be done. That cover crops example in terms of biomass inputs was actually about 3.5 tonnes per hectare greater input under that treatment when you count both the cover crop itself and the extra yield that came as a result of that treatment. So I'm conscious of time, I want to wrap up. Drylands, it's not just an Australian problem. I do try to have an eye beyond just Australia. Water availability and increasing lack thereof is going to be a major problem for all of this. Conservative nitrogen management, I think, is a real issue here. And we do need to understand losses more, particularly in the context of how much the stuff now costs, but nonetheless. Ultimately, these are going to limit how much you can grow and therefore how much carbon comes in. And increasing net primary productivity really is the key. Again, looking at those main points. So I discussed a little bit of that new knowledge right at the start, and I'm going to take us back there. Now, what does all that I talked about far earlier on in the talk, with how we measure organic matter, what it actually is? What does that mean for us trying to manage it out in the paddock? Well, first of all, the thing to remember, it's stabilised, but mostly not stable. One of the reasons why no-till has been suggested as being so important for at least arresting the decline, if not an outright increase, achieving an outright increase in organic matter. Much carbon is lost during decomposition. That's not up for debate, that's clearly a matter of fact. And it's worth bearing in mind that a reversed good practice can see a decline quite quickly. Interestingly, and 2 weeks ago, that global change biology paper came out. It's starting to be clear we do need to question the absolutism around the eye of the needle principle. So maybe not all stable organic matter went through a microbe first. And that does have implications when we're thinking about strategies that focus mainly on microbial stabilization. So where there's been a lot of work looking very carefully at stoichiometry, notwithstanding the difficulty of actually getting the carbon, the nitrogen, the phosphorus, the sulfur, the clay it's going to be stabilized to, and a willing microbe all in the same couple of microns of soil, of space. Nonetheless, really it comes back to actually feeding the system and feeding the crop. And with that, I'd like to thank you.
Thank you, Mark. Perfect timing. Amazing. Last slide.
That's all right. I can...
Sorry.
Yeah, that's all right. The last slide is first for some reason. There we are.
I have to follow the protocol. Mark, thank you for your in-depth and very elegant presentation on the fundamental aspect and also the applied as practical aspect of soil organic matter. Now we got another 15 minutes for some discussion. I strongly encourage young, budding scientists to ask questions. UWA is doing a lot of work on soil organic carbon, like CSIRO or any other organization. So please ask a question. Just tell your name and then ask.
Hi.
Rob Harrison. So you didn't speak much about like free living or free fixing rhizobia or bacteria in soil then. How important are they? And are they like, I've read some studies that they're not even important in the system, and some others would say they're important up to 20%.
Well, you just covered the range. That's definitely an area of research that is underdone, I think it's clear to say, and underdone in Australian systems. There are plenty of natural systems where N by free living N fixes is actually probably the dominant form of input. But they're very arid, slow-growing systems from which we don't export anything from. The fundamental thing is the sheer amount of nitrogen that is harvested every year, because that's what we're growing this stuff for carbohydrates and protein. Protein's a lot of nitrogen in it, and of course we're harvesting it and taking it away. I don't think that we would ever get close to them being, to free-living N fixes being enough to supply a productive cropping system. And another complication there is, as you're probably aware, of course, free-living N fixes, well, N fixes full stop, tend to get a bit lazy when there's lots of available nitrogen around. So it's not really it's something that would be quite difficult to manage to get them to do their best and then top it up. So definitely room for more research there, I would argue, because you're right, somewhere from 0.2 to 20 is rather a lot of orders of magnitude for us to have uncertainty on, but at the same time, I think it would be pragmatically, for the same reason that I sadly, for somebody who has the odd distinction of having a PhD in composting, dismissing organic amendments in this question.
Thank you. Any other questions?
Hi, Mark. From UWA. In one of your slides, I came with this, I had a meeting for it. You mentioned that the root system of the crops.
Yep.
In fact, what we have been doing in Australia but elsewhere last 5 to 10 decades, we have been increasing the root system.
Mm-hmm.
So if you look at the modern wheat,
Yeah.
modern take crops, whether it's in Australia, China, all those places, coincidentally, the plant breeders have been correcting
Yeah.
that they use in the harvest index, and we have halved the
Indeed.
dry matter, woodland density, and became more efficient in taking nutrients and water. So that idea, if we start breeding for better root system, we may have the permanent compensation.
Yes. Yeah, I mean, ultimately, the upper limit is how much carbon the crop can fix, and then the next question is where it puts it, I guess.
Perhaps
Yeah.
better row orientation, better seeding rates, and other
Yeah.
things may give us the same in a different way, agronomic way.
Yeah.
The second point is the cow crops, again, a big issue you mentioned, because of the probabilities of summer rainfall in our Mediterranean environment, and a lot of studies done all the years, the probabilities of successfully raising a cow crop during summer is marginal. The second point is that the farmer doesn't have the ability to store summer crop as a tactical decision when they get summer rain. So two things. One is that the summer rainfall is not enough to sustain continued growth and it takes the water. The second is they can't store some crops or pastures in the system because many years they have to wait.
So those were the things that sort of sat over the top of us when we did this work. And look, I will be upfront. It was funded through lawn care, through a lawn care program, done on the back of on the sniff of an oily rag, basically to try and have 20 sites run for it, principally as demonstration trials to see whether or not it could work. What surprised me actually is that it wasn't just 1 or 2 sites where it could work. There was a pretty even split across those rainfall areas. Clearly, even in the wettest areas, you might not get the rainfall and it can send you backwards. But I think the really important message I got out of that project, and it was running collaboration with a lot of grower groups, so definitely had buy-in from them on what I'm about to say here, is that ultimately the answer isn't yes, but it also isn't no. It is very context specific. There can be exactly almost the mundane practicalities of storing the seed, the opportunity. Conversely, at least one of our sites was sown from an aircraft, which, if you have access to it, all of a sudden it rains, there's a rain coming, out you go with the seed, which is literally what he did, and they got good germination. from it, and that was the site where the header, header lost its GPS connection. So I don't have good yield data from 2 of the 4 plots there. But going off the 2 plots, they followed the same trend as the Kangaroo Island site I did show you. So there are definitely barriers to it. It's definitely not an all-in like it is shown in wetter areas, but it's also not completely no either, but it will take some care in managing.
Mark, UWA is doing a lot of work on root dynamics.
Yep.
Plenty of work, and as Civic has indicated, the root cause
Yes.
of soil organic matters roots.
Yes.
So I think, you know.
And root exudates in particular.
Is there any other question? Thank you, Sudhir. Yes, please. Farrell.
We've got a much more fundamental question. Right back at the start, the definition of soil organic matter.
Uh-huh.
And I have wondered, to me, soil organic matter is a lot more than what you've described. Like you don't include the water extractable organic matter, which is all organic matter fraction. And I don't think we've touched the definition. What do you call the precursors of the stabilized material that's now protected from decomposition? And the reason I ask this is this a functional thing because it's easier to quantify if you're trying to look at organic matter accumulation? And does that then constrain how you might mechanistically overcome some of the problems which you
Yep.
alluded to?
So on where the DOM, the dissolved organic matter, is in the fractionation scheme that we do, it ends up in with the humus-like fraction, the mineral-associated fraction. Simply it's size fractionation. Of course, the water-soluble stuff ends up in the bottom too. I can use the hackneyed argument that it's normally less than 1% and therefore it doesn't matter. That's a very, very poor argument and I'm not going to. It might be less than 1%, but quite a lot of carbon goes through that little pool. An analogy some people may be aware of, done a lot of work on organic nitrogen in the past. There are very few, there's a very, very small pool of, standing pool of amino acids in the soil. Doesn't mean they're not important. The reason there's very few of them there is because they are important the moment they're produced. They're taken up by something. Now, DRC is a bit more complicated. Not all of it. Some of it just happens to be soluble, but isn't actually particularly usable at all. In soils, I find DLC equally fraught with being operationally defined in some regards because of that. It's which bit of it is actually important, and having a tool that really picks that apart. Particularly when you then get into the aquatic space, where I would get shot if I said that humic acids don't exist. So. Yeah, there is some work. There's lots of work in the space of trying to understand and model this, and there's some really interesting space looking at it from a fluxes perspective rather than a pools perspective, and the pools actually being artifacts of those fluxes rather than being dominated thing. One thing to bear in mind about our fractionation scheme, whilst we've tried to make it as ecologically relevant as possible, it has been calibrated to Rothc and therefore also sits underneath our national carbon accounts in this country, which is a big differentiator between it and a lot of other schemes. And that was from a huge set of data 20 odd years ago when Jan led that work. But nonetheless, we've always had to have an eye as to how this can be operationalized at scale, because the perfect scheme that takes 3 weeks to get your answer for one sample is of no use whatsoever, really, beyond understanding the fundamental processes. So yeah, there is quite a lot of work to do still in that space. I'm pretty comfortable in defending our scheme to most comers and have done in international conferences too, and the fact that I think we have a good balance between operationally achievable and ecologically relevant, but it's definitely not completely right.
Our group is doing a lot of work on stabilized
Yes.
work. I have to follow strictly the protocol. 2 questions, one from you, and probably 3 questions Louis there, and from some budding young PhD student. Yes, please.
Thank you. Where's Laura? Access Tech. Great presentation, thank you. Early on in the presentation, you were talking about some very specific measurement techniques.
Yep.
IR,
Right. You're quite right there. That's
Couldn't really draw the link
sorry about that. And
high precision,
yep.
sciencey bit and the broad acre.
I suspect you probably already know, but the bit between them is something called mid-infrared technology that allows us to rapidly, so that fractionation process, just as an operational thing in the lab, you'd be looking at getting maybe 10 samples a week through our lab, clearly not operationalizable. But one of the big outcomes from that National Carbon Project that Jeff led 12 years ago was really being able to calibrate rapid measurement techniques such as mid-infrared to it. So instead of 10 samples a week, you're looking at 300 or 400 samples a day of estimates for it. That's how we do it, still being fairly bench-based group. There are other groups within and external to CSIRO and many companies working on doing this in the field, either with mid or near infrared and a mixture of other sensors as well to correct for things like moisture, gravel content and things like that. I'm not in the sensor development space and generally do sit in the biogeochemistry process space, although I also clearly do more than just dabble in agronomy. But the two, actually doing it at scale, there are definitely developing tools that allow both rapid estimates on the spot. And the other goal of that soil organic carbon monitoring project that I mentioned is actually to get a baseline gold standard set of samples and data that will be publicly available for companies to use to develop tools. And those samples will be in the National Soil Archive and accessible by anybody with a good reason for it. And the data will be, the underlying data for them will be publicly available. A deliverable of that project is that happens at the end of it, regardless of whether or not we've written our papers. There will be no embargo.
Thank you, Mark. Louise?
So Mark, carbon and nitrogen are so intricately linked.
Yep.
Often known soil scientists put themselves in one spot or in the other, and I'm definitely one of those people who tend to focus on nitrogen losses. And in our environment, we find that we often get a lot of losses in response to the fact that we get we have more nitrification happening in comparison to, say, mobilisation of nitrogen. And it's often considered that if you start increasing your soil organic carbon or add organic matter to the soil, that you might reverse that situation. But work done by Farrell showed that that wasn't the case.
Yep.
And that was backed up by work that Louise Fisk did.
Yes.
But what Louise Fisk did show is that when you started adding synthetic root exudates to the soil, you might be in there with a chance.
Yeah.
Hence the sort of the why cover crops in another sense seem so appealing, but not necessarily practical, as Cannonbot pointed out.
Yeah.
Is there any other way we can manipulate the system without having to introduce a plant into it so that we can get those root exudates without a plant?
The trouble is it's not necessarily the plant is the perfect delivery system for them, and that's the real kicker. So the other thing to bear in mind is some of these cover crop studies don't actually twig the fact that the plant stopped photosynthesizing quite some time before you harvested it. Your period of a living plant is actually much shorter than you think it is in these systems. I can't conceive of a way you couldn't spread it on the surface because it won't go in and it'll get eaten at the surface. I mean, these a lot of these compounds we're talking about being turned over in matters of minutes, if not less than that, certainly hours. They are probably only about 1 or 2% of the dissolved organic carbon pool at any one point in time, but the flux through them is absolutely massive. Short of something that puts roots in the soil without actually being a plant, I cannot see a way around that.
Just the final question, because I will follow strictly the protocol. Final.
Be a rebel, Nancy.
I just had to say that one, given the time, just
No. So the cover crops were, sorry, I should have mentioned that. We terminated the cover crops well before seed set. There are grazing options for cover crops, although don't forget, if you graze them, you're taking away a load of that carbon in the process. Sorry, carry on. Yep. Okay.
We have, Professor Lynn Abbott has brought a lot of food. We have plenty of time to go on and more discussion. Now, unless there is any burning issues from PhD students, any questions? Okay. Thank you, Mark. Would you please join me in thanking Dr. Mark Farrell for fascinating and